assignment, and the first thing we want to do is distinguish the two retroviral oncogenes that we are learning about for our first exam, and they are RAS and MYC, and RAS is a monomeric G protein we learned a lot about, and we talked about MYC some already, and we know it's a transcription factor. So let's distinguish them with regard to the type of mutations they can have and which ones lead to cancer and which ones don't. So that'll be our RAS gene. That will be our MYC gene. And these could be, and you could label these. I don't bother to label them, but you could, and you'll have to do that well on the exam, right? Okay. And so, like with any gene, there are really just two types of mutations that can happen. We can affect the product of the gene by having mutations in the transcription unit, or we can affect the expression of the gene by having mutations in the cis regulatory region, right? So regulation. And we picked these genes. All this stuff is getting picked for specific reasons, and this specific reason here is that it is the product of RAS that is the problem, and we know that really well, right? That this is a protein coding gene. It codes for a protein. There's importantly, the coding region. So we're not just going to affect the RNA product. We're going to affect the RAS protein that's produced, and it's going to be defective, and we know how in detail. So that's the only way that this gene can contribute to cancer, is mutations in the coding region of the transcription unit. Okay? So it's nothing about where, when, or how much of the gene gets expressed. That's what the cis regulatory region controls, and it's only those three things. Now we're turning our attention to MYC, and in MYC, there's only one place we can have mutation, and it's not the transcription unit. When MYC is involved in cancer, it's the normal wild-type MYC protein. And we've seen that when the body is making MYC, it's worried. So the body tries to block transcription. The body tries to degrade the mRNA. The body tries to block the mRNA from being translated by the ribosome. Then the body tries to take that MYC protein and break it down really fast. And so what we're going to do is we're going to cause mutations here that are going to lead to way too much MYC protein. And so these downstream attempts to reduce it won't be enough, won't fast enough. This is all about making mutations in the MYC oncogene cis regulatory region. And the way this occurs is typically there's going to be a chromosomal translocation. It's like the woman had with cancer, but it's just with the skin cancer, but it's just different genes. In this case, the chromosomal translocation that we're going to learn about is going to be in a blood cell, a kind of blood cell that's called a B cell. Sam came in and taught us a little bit about T cell therapy, CAR T cell therapy. This is a different kind of blood cell called a B cell, and that's going to become cancerous. All right, so that's where we're at, and that's the first super important concept that we need to cover, okay? All right, so then on to this. All we're going to do is draw oncogenic retrovirus, and there it is. It's that same drawing, and just have to do this to the drawing this time. We've already drawn it for assignment, right? We're going to draw it again, and one thing we're going to do is we're going to talk about this. MYC is going to be the oncogene in there. And and you have to add this to the label. In this case now, it's that MYC transcription unit. And so where is the transcription unit? Where is its cis regulatory region? It's not here. This is going to be the new cis regulatory region. We're making a hybrid gene, and we're doing that just like the woman did with skin cancer, but this is not the body's a chromosomal translocation, right? This is a rearrangement in the virus and the viral genome that made a mutation in the viral genome by adding, by accident, an extra gene, and it's a host gene. And The thing you need to know here is that when a virus infects a cell, the virus is just trying to copy itself. It's trying to replicate as much as it can. So these cis regulatory regions in the virus, so that's the cis regulatory region of the virus. And what are these like? Well, they regulate where, when, and how much. Always. That's what regulatory regions always regulate, and only that. And for a virus, where? Yeah, everywhere. When? All the time. How much? As much as possible. Okay? So what's happening here is we just accidentally added this transcription unit to this virus, and when it gets infected, when it affects the next cell, it's going to try to make as much of this as possible. And it simply overwhelms those downstream efforts to minimize the MYC production. Yep, that's the first one. So first assignment, draw and label the genes in an oncogenic retrovirus with MYC. Okay, and so that's your first drawing. Take that, make that drawing and send me a pic. All right, so then here comes the next one. The next one is that in addition to oncogenic retrovirus, we have a second example of overexpression of MYC that leads to cancer. And this is our example of a chromosomal translocation, and it's a type of cancer called Burkitt's lymphoma. Lymphomas are a type of cancer in the blood, so we could have solid tumors like a kidney tumor or a stomach tumor or a breast cancer tumor or a brain cancer tumor like that. These are tumors of the blood, and so we just have too many blood cells. And in this case, the blood cell that we have too much of is a B cell. And B cells' job is to make a protein that's called an antibody. And that's all we need to know, is that these cells are going to try to make lots of antibody proteins. And there are a couple of different genes for different parts of the antibody protein. There's a heavy chain gene and a light chain gene. And so here's the heavy chain gene, is it, for example, and this has a very strong, so it's an antibody gene, and it has a very strong regulatory region. So where, when, how much of this gene to make. And the where is in the B cell, and the when is as the B cells are maturing, and the amount is this problem, and it's lots. It's a specialized cell that's job is to make antibody, so it makes a lot. It has a very strong cis regulatory region, and that's going to be what gets joined together with the MYC. We're going to have a chromosomal translocation that's going to bring together these two to make a hybrid gene. And that's going to be your second drawing, is to draw the two genes and show how they become a hybrid gene. Is this an oncogenic retrovirus? No, this is in just the patient's normal body, and this is an example of a chromosomal translocation where we bring one cis regulatory region together with the wrong transcription unit, and in this case we make way too much of it. MYC is normally involved in stimulating the cell proliferation of in our blood cells. So it's also a cell that's vulnerable to having too much MYC. Okay, so Burkitt's lymphoma is a rare type of cancer. It's called a non-Hodgkin lymphoma, and these kind of details are not the most important part of our course, right? It's a course in molecular biology, and we've already described why we're using this as an example. That's the most important thing. Just want to say what it is so you have some context. It's typically in children, and it's very rare. These are non-Hodgkin lymphoma types, and down here is ours. Rare, but unfortunately very aggressive, which means that the cells divide really fast. A person who has Burkitt's lymphoma might only survive for a couple of months. Yep, maybe longer, but it underscores the problem, is the MYC is being overexpressed, and MYC is really good at pushing cells down the cell cycle. Let's just draw that out because we've talked about it in class. And that is, here's a gene, and here's MYC. And if MYC gets a chance to do its job, it's going to activate several genes, and the one we'll focus on is a one that's going to code for a cyclin protein. And cyclins are a family of genes. They have different letter names, cyclin A, B, C, D, E, like that. And we're going to make cyclin, and we're going to push it through the cell cycle over and over again. There's not going to be a red light when the cell comes up to it. It's going to be green, green, green. Okay, so that's what's going to happen here, and it's going to be too fast. So the MYC gene plays a central role. So this kind of cancer has been described for years by doctors, and now we know this is the trouble. Translocation and overexpression. So just read through that, and then here's the translocation. This is chromosome number 8. This is chromosome number 14. Human chromosomes are named by numbering them just based on the longest to the shortest. Chromosome 1 has the longest, is the longest chromosome, and chromosome 22 is the shortest. And along their lengths, different chromosomes have different genes. The chromosome 8 is where we find our MYC gene, and it's c-MYC, cellular MYC, because it's not a retrovirus story. That would be a v-MYC, and it's going to get translated to chromosome 14, where we have the gene for human antibody heavy chain. And so they're going to get joined together. That would be the cis-regulatory region. That would be the MYC. And the next thing to say is that we can see this under a microscope. So scientists have originally, probably back in the 1950s, characterized Burkitt's lymphoma by looking at chromosomes under a microscope slide like this. And when scientists do that, we need to know this term. It's a karyotype. So they spread the chromosomes out and they look at them, and they see the chromosomes are lined up based on their length, and they're not putting in all the numbers, but there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 14, like that. And what you can see is that it's a pair. One is from mom, one's from dad. We can't tell which. And in this case, you can see that one of the chromosomes is shorter than the other in both these cases. And so let's just go back to here and say, why can you see that? And the reason you can see that is this drawing is imperfect. Let me look at this for one more second. So what this should show is normally this chromosome is just a bit longer here than that one. And so when we have a chromosomal translocation, chromosome 14 is actually going to be looking a little bit longer, and chromosome 8 is going to be a little bit shorter. And you can see this designation here. They're saying that it's a little bit shorter and a little longer. Nothing to do with the genes, just the happenstance length of the chromosome beyond the gene. And that's what the doctors and scientists could see is the difference. They couldn't tell what the genes were. They really didn't know what the genes were at all until we could analyze the DNA in more detail. But even back in the 1950s, they were able to see this chromosome gene length difference. And so this assignment is to draw the two chromosomes and show how, like on the quiz, here's this chromosome, label them, with the parts, and there's MYC, and there's the antibody gene, and then you're going to circle the antibody gene, regulatory region, got connected to the MYC. So that's our second drawing that you're going to label and send a picture to me too. And it's just like, I think it was question 5 on quiz 3, only it's a different story of chromosomal translocations. Different chromosomes because it's different genes. Yep. And so this is just an image of what that's like. So we have the antibody or immunoglobulin heavy chain enhancer, regulatory region, and it's just blasting out lots of this MYC protein in Burkitt's lymphoma. So that's the second one. Draw and label the genes involved in the chromosomal location and show what the effect is of the chromosomal location, translocation. Great. So all types of Burkitt lymphoma are characterized by the dysregulation of c-MYC by one of 3 chromosomal translocations, because I mentioned that there are multiple genes that are part of the antibody gene. So 3 different chromosomal translocations, all of them involve the overexpression of MYC, normal MYC protein, in these B cells. Yep. All right. So then, luckily, there are treatments for Burkitt's lymphoma, and it's a cancer with a really good treatment. and cure rates. And I glanced, they don't really have pharmaceutical, like specialized chemicals like tamoxifen we saw in breast cancer. I think they just do kind of traditional types of chemotherapy, and they're successful at really curing folks that have this kind of cancer. So really dangerous and fatal cancer, very rapid, but not if we can get treatment quickly like that. Okay, so then I want to just insert here, because we're going to go on to our last drawing. At the end of this chapter, there's some, at chapter 16, there's some slides like this that show and introduce the complexities of these pathways and how they're used in specific cells in different examples at different times and so forth. And we don't need to dive into the details of this. We're diving into the details of this by this thing of the 3 examples where signal transduction pathways are involved in amplification, integration, and then this variety. The same signal transduction pathway occurs again and again, but activated by different receptors in different cells and leading to different responses in those different cellular contexts. So we have lots of different signal transduction pathways, but they actually are repeated and used repeatedly so that there aren't as many as you might think. And so this just is introducing you to a little bit of it. Sam showed that in that CAR T cell thing. He showed, well, look at the blizzard of pathways, right? But they can be sorted out and understood, one at a time. Okay. Another figure I wanted to show you near the end of the chapter is just, I think it's near the end of the chapter, but it just shows that actually this RAF-MEK-ERK pathway, so we learned about the MAP kinases, they're actually all held together by this protein that's called a scaffold protein. And that's cute to see just because they're trying to find the downstream targets, like that. And so why not just hold them together and let this occur and its amplification. So just a couple of slides from our end of a chapter or more details in our chapter to emphasize, the importance of this chapter in our course and how we want to understand the details of a few signal transduction pathways, and then and then also an appreciation of the overall purpose of these pathways to amplify, integrate, and provide a variety of contexts. So I also want to show that just not a key concept here, but another signaling pathway in this textbook is the Wnt beta-catenin signaling pathway, and that can also activate MYC. So just a little taste of how these have flexibility. To help you learn these, I've identified some really classic pathways, like we're able to go from the PDGF released by platelets under a scab all the way to the activation of the cell cycle by having MYC get made and then bind to the cis-regulatory region of a cyclin gene, and the cyclin gene gets made into cyclin proteins, and that's what's needed for the light to turn green and the cell cycle to advance. Okay, so let's go on to our third drawing, and this is a new term for us. It's called gene amplification, and when doctors look at certain cancers, there are some cancers where a certain gene gets amplified. So this could be our MYC gene, and our MYC gene, and our MYC gene. And this is just in that one cancer. And what's happening here is gene duplication, and it's the same concept as we've already learned about, but a different setting. This will not lead to new paralogs. It's not occurring in the cells that could make sperm and egg and send genes on to the next generation. So this is not about evolution. This is about the problem in this cancer patient. And what happened is they make too much MYC by having the normal MYC gene, but just copying it many, many times by accident through the gene duplication. And when people have this, they could have it be MYC or some other gene. There are other oncogenes that could have occurred gene amplification, and we get too much of them. What's going on here? This must be a story of the cis-regulatory region. They're not changing the product, they're making too much of it. They're making where, when, how much. And here they're making too much just by having too many copies. Gene duplication is really rare, but cancers get out of control by just giving up on things like repairing DNA damage. And so things like gene duplication and mutations get higher in cancer. And when you see this in a cancer patient, they might have 100 copies of the MYC gene that have been copied that many times. Yeah. So that's going to be our last drawing, and it's just going to be to draw the MYC gene several times in a row and show that that's going to make, 5 times in a row means 5 times more MYC in general. And that's our third drawing. And here's MYC. MYC gene amplification is often acquired in lethal, distant breast cancer metastases of unamplified primary tumors. So one thing that happens is these can occur in other cancers as well, not just in the blood cells, where MYC is important for stimulating the cell cycle in normal cells. And so we see that these that cancer is typically mutations in multiple genes, and you have multiple problems that just keep adding up, and the cancer gets worse and worse and worse. And in this case, it's just that we are going to make too much MYC by making 3 times more MYC or 5 times more MYC. And so that's your last drawing, and with that, you're set with this. And those are our 3 drawings in this assignment, and they all loop back to this. So we have to be, in this case, in the context of our RAS and our MAP kinase pathway, like occurs under the skin, in the skin at wound healing, or in other settings too. Yeah. And then also remember that this is not the only way MYC can get activated. This is not the only way RAS plays a role. These are just, this is a example of a nice, clean, relatively simple story that takes us all the way through from a signaling molecule to a cellular response. The signaling molecule is platelet-derived growth factor, and the cellular response is stimulate the cell cycle. Growth factors stimulate the cell cycle, yay. In general, and like I said, Sam showed the blizzard of signaling transduction pathways that are occurring in that, activating that T cell. That's what signaling signal transduction pathways usually look like. Lots and lots of different stories of amplification and integration and a variety of uses. Yeah, so this is a great one for us to really focus on. And again, that PDGF a video that we just did our assignment on and our quiz 3 on, it's a central part of our course, like the original videos were. Transcription regulation, and now this is receptors and signal transduction pathways and cellular responses. So with that, we're all set for this, and so that's this video. Yep.